Good morning, everyone. I'm Rashi Degi from BSc Medical Biotech, and I'm going to give a presentation on the topic of dendrimer. Dendrimer are nano-sized symmetrical molecule in which a small atom or group of atom is surrounded by the symmetric branches known as dendrons. The structure of dendrimer possesses the greater impact on their physical and chemical properties. They grow outward from the core shell, which further react with hormones having one reactive or two dormant molecules. Dendrimer unique characteristics such as hyperbranching, well-defined spherical structure, and high compatibility with the biological system are responsible for their wide range of application, including medical and biomedical areas. Particularly, the dendrimer three-dimensional structure can incorporate a wide variety of drugs to form biologically active drug conjugated. In the review, we focus on the synthesized mechanism of drug encapsulation in dendrimer and their wide application in drug history. Newcomb's group independently reported synthesis of similar macromolecule named as arboreal. In 1985, Donald A. Tomalia synthesized the first family of dendrimers. In 1978, Fritz Bocktel synthesized the first cascada molecule. Here are the type of dendrimer. The mechanism of dendrimer are particularly attractive as they offer a high drug loading capacity. Method of dendrimer drug delivery are encapsulation of drug, and the second is dendrimer drug conjugating. Encapsulation of drug, interaction between the dendrimer and the drug to trap the drug inside the dendrimer. Such system can be used to encapsulate drug and provide controlled delivery. Example, DNA was complex with PAM-AM dendrimer for gene delivery application, and hydrophobic drug and dye molecule were incorporated into various dendrimers. And the second is drug conjugated. In dendrimer, drug conjugated, the drug is attached through a covalent bond either directly or linker spacer to the surface group of dendrimer. Dendrimer have been conjugated to various biologically active molecules such as drug, antibodies, sugar, molecule, and lipid. The drug loading can be done by varying the generation number of dendrimer. Release of the drug can be controlled by incorporating degradable linkage between the drug and dendrimer. Conjugated of PAM-AM dendrimer with cisplatin, a protein anti-cancer drug with non-specific toxicity and poor water solubility. The conjugated shows increased solubility, increased systematic toxicity, and selective accumulation in solitude. Ideal property of dendrimer are inert and non-toxic, biodegradable, non-immunogenic, able to access barriers such as intestine, blood tissue barrier, cell membrane, etc. Able to stay in circulation for the time needed to have a clinical effect, able to target to specific structure compatible with the guest molecule. Must protect the drug until it reaches to the desired site of reaction and release.
Good morning, my name is Ritu Gautam and I want to present my presentation on the topic of liposomes in drug delivery. So let's start this presentation. There is an introduction of liposomes in drug delivery. A liposomes in spherical vesicles having at least one lipid bilayer. The liposomes can be used as a drug delivery vesicle administration of nutrient and pharmaceutical drugs such as lipid nanoparticles in mRNA vaccines and DNA vaccines. Liposomes can be prepared by disrupting biological membranes such as by sonication. Liposomes are most often composed of phospholipids, especially phosphodietycholine. May also include afterlipids such as phosphate diethyl ethanol amine so long as they are compatible with lipid bilayer structure. The size of liposomes range from 20 nm up to several micrometer. Liposomes derived from Greek word which is lipo means fatty constitution and some means structure. Liposomes were first described by British hematologist Glottis Alec D. Bengham 1961 published 1964 at Brahmin Institute in Cambridge. So there is a structure of liposomes for drug delivery. So as we shown in this picture, there is a DNA and a hormone peptide, lipid soluble drug in bilayer, lipid bilayer, drug crystallization in aqueous fluid, protective layer against immune destruction. Liposomes are composed structure made of phospholipids and may also contain small amount of other molecules. Though liposomes can vary in size from low micrometer range to tens of micrometer, unilamellar liposomes, as pictured here, as typically in the lower size, range with various targeting ligand attached to their surface, allowing for their surface attachment and accumulation in pathophysiological area of treatment of disease. The main component of liposomes are phospholipids and cholesterol, which is shown in figure. This is a structure of phospholipid which contain hydrophilic polar head and hydrophobic tail. Phospholipids are the molecular building block for the liposomes, and it also consists amphiphilic molecules are consist of hydrophilic polar head and hydrophobic tail. Hence, the affinity for hydrophilic drug can be encapsulated in the aqueous phase, and hydrophobic drug molecules can be incorporated in lipid bilayer. There is a structure of phospholipid and liposomes. In the left hand side, there is a structure of liposomes, and right hand side, it is a structure of phospholipid. As we shown on this picture, detect that these are the liposomes, and which is shown hydrophobic drug which is red in color and hydrophilic drug which is green in color. It also consists hydrophilic head and hydrophilic tail. This is the structure of phospholipid, and let's discuss about the classification of liposomes. These are divided in the three classification, which is based on the structure, based on the composition and application, and based on the preparation. So there is the first based on the structural parameter, MLB, multilamellar vesicles, OLB, oligonucleotide, oligo vesicles, and UB, UB and MMB, and UB also consists further divided and MDB and SUB, which is and GUB and LUV. GUV gained unilamellar vesicles and LUV larger unilamellar vesicles. And now this is a second based on the composition application. This is further divided in this which is CL constitutional liposomes, which consist neutral and negatively charged phospholipids and cholesterol, and fusogenic liposomes. It consists RSVE reconstituted Sandel virus, and pH sensitive liposomes using phospholipids such as PA or doped with GA. Catenoic liposomes, catenoic lipid with dope, long circulatory sleeve liposomes, high TC made using cholesterol and 5 to 10%, immunoliposomes which are attached to mononuclear antibodies. There is a third based on the method of preparation, DRV, dehydration rehydration method, REB, SUV and OLV made by reserve phase evaporation method. MLV and REV. MLV made by reserve phase evaporation. BET vesicles prepared by induction technique. FAT MLV frozen and thawed MLB. SPLB stable plurilamellar vesicles. These are the method of liposomes preparation. It consists further divided into two method techniques, and passive loading technique and active loading technique. Passive loading technique further divided into three methods. First is mechanical depression method and solvent depression. Then third one is detergent removal method. Mechanical depression method further divided into different methods and process: lipid film hydration by hand shaking and non hand shaking and freeze drying, micro emulsification, sonication, French pressure cell, membrane extraction, dried reconstituted vesicles, freeze thawed liposomes. The second one, solvent depression, ethanol injection, either injection, double emulsion vesicles, stable plurilamellar vesicles, reserve phase evaporation vesicles. And third one, detergent removal method, which consists detergent, colloid, alkali glycoside, tritone X100, removal from mixed missiles by dialysis, column and chromatography and dilution. Now these are the choice of liposomes preparation method depend on the following parameter: the physiochemical characteristics of the material to be interrupted and those of the liposomes ingredient, the nature of medium in which the lipid vesicles are dispersed, the effective concentration of interrupt substance and its potential toxicity, additional process involved during application and delivery of the vesicles, optimized size, polydispersity and shell life of the vesicles for the intended application, and also batch to batch reproducibility and possibility of large scale production of safe and efficient liposomes products. There is a mechanisms of action of liposomes in drug delivery system. A liposomes consist of a region of aqueous solution inside a hydrophobic membrane, and hydrophobic chemical can be easily dissolved in the lipid membrane, and this way liposomes are able to carry both hydrophilic and hydrophobic molecules. Steps involved in liposomes action of drug delivery. There are four steps. First is adsorption. Adsorption of liposomes to cell membrane causes its contact on the cell membrane. And second one, endocytosis, adsorption of liposomes on the cell surface membrane followed by engulfment and internalization into the liposome. And third one is fusion of lipid bilayer of liposomes with the lipodial cell membrane by lateral diffusion and intermingling of lipids result in direct delivery of liposomes patient in the cytoplasm. And second is lipid exchange. Due to the similarity of liposomes lipid membrane with cell membrane, phospholipids lipid transfer protein in the cell membrane easily recognize liposomes and cause lipid exchange. And, for example, in case of cancer cell, they consume large amount of fats to fill the requirement of rapid growth. They recognize the liposomes loaded with anti-cancer drug as a potential source of nutrition. When they are targeted by liposomes, they get absorbed. Once the anti-cancer drugs are released from the liposomes into the site, cancer cells are killed by this drug. There is the advantage of liposomes: improvement and control over pharmacokinetics and pharmacodynamics. Second one is decreased toxicity. Third, enhanced drug activity against intracellular pathogens. Fourth, liposomes used as a target selective. Fifth, enhanced activity against extracellular pathogens. There is advantage of liposomes: first, sterilization; second, short shell life and stability; encapsulation efficacy; removal from circulation by the reticuloendothelial system (RES). These are therapeutic application of liposomes. First, site avoidance delivery, site specific targeting, intracellular drug delivery, sustained release drug delivery, intraperitoneal administration, immunological adjuvant in vaccine. These are the therapeutic application of liposomes. Thank you.